Our body is made up of cells. As our body undergoes constant wear and tear, we need new cells to help us replace the old, dead, and damaged cells. We also need new cells to help us grow and develop. And to do this, our cells undergo something called mitosis. It is a form of asexual reproduction that helps us replenish cells and grow. This is mediated by a process called the cell cycle that regulates cell division and contains vital checkpoints to ensure that the cell and the resulting daughter cells are healthy and can function normally. So now let's take a look at the process itself. In eukaryotes, the cell cycle can be divided into 3 states: interphase, resting, and mitosis. Since it is such a complicated process, a process called the cell cycle control system monitors and dictates the progression of the cell through the cell cycle. Positive and negative regulators monitor, halt, or advance a cell through 3 major checkpoints to ensure that the cell can create healthy daughter cells. Let's first take a look at interphase. Interphase is basically the period of time that a cell does anything other than mitosis, and by intuition, cells spend most of their time in this state. In this state, a cell prepares for mitosis by increasing in size and duplicating its contents/DNA. The interphase state consists of 3 different phases, namely Gap1 or G1, synthesis or S, and Gap2 or G2. Let's look in detail at each phase. First, let's pick a cell. Any cell. There. Now let's give her a name. Let's say Abigail. Nice. We will now focus on what happens inside Abigail as she progresses through the cell cycle. Abigail currently is in the phase G1. During G1, Abigail is metabolically active, doing her role in her local community of cells and continuously growing. Now, understand that in the microcosmos, a single-celled organism relies on diffusion to help transfer materials inside and outside of its membrane in an effortless way. The greater the surface area of the membrane, the more things can diffuse inside and out. However, since according to the square-cube law, as an object gets bigger, the volume increases faster than the surface area. Thus, the larger an organism is, the harder it is to maintain homeostasis, as the diffusion rates are not enough for it to supply itself with nutrients and dispose of waste. And thus, when Abigail purposely gets bigger, it may seem counterintuitive, but remember that she is preparing for division. Now that Abigail is bigger, she faces a challenge. She now needs to proceed to the next phase, synthesis. However, there are a few problems. She first asks a few questions. Is she large enough to divide? The protein retinoblastoma, or RB, helps solve this problem. RB is a negative regulator that will inhibit progress if conditions are not met. In an active state, RB binds to transcription factors, which are chemicals that help activate sections of the genome to begin the production of proteins that promote the transition from G1 to S. As the cell slowly gets bigger, RB gets phosphorylated until it becomes inactive, releasing the transcription factors to now produce proteins from the DNA needed for the S phase. There are 2 more proteins, P107 and P130, that do a similar function as RB, including determining if there are enough nutrients and if growth factors from neighboring cells are present. These 2, along with RB, make up the pocket proteins, which are all negative regulators. By the way, it is worth to mention that another negative regulator, P27, acts in a similar manner as RB, and as more and more cyclins are produced, will become inhibited and lets the cell cycle continue. Next, P53 comes to the act. P53 determines if Abigail has sustained any damage in her DNA, for example, from UV light exposure. If P53 detects that there is DNA damage, it will signal for enzymes to help repair the damage and will also increase the production of P53 itself. As P53 levels rise, this triggers the production of P16. This inhibitory regulator acts when there is damage in the DNA and will disrupt CDK cyclin complexes and lead to the release of the integrated P21 in the CDK cyclin complexes, which in turn will trigger the dephosphorization of RB and will inhibit the transition into the S phase. If P53's efforts to repair Abigail's DNA are not successful, then it will signal for apoptosis. Note the fact that all negative inhibitors, which include but are not limited to RB, the PXX inhibitors, and more, are all proteins and they are encoded by the specific genes referred to as the tumor suppressor genes. These proteins are all constructed by the protein synthesis cycle as per usual. If some of the less strict negative regulator requirements are not met, For example, ample nutrients, little stress, or of the like, then RB or another inhibitor may force the cell into the state resting, or G0. In other cases, the cell receives molecular signals from its surroundings to differentiate into a specific type of cell for a specific type of job, and will also enter G0. In G0 state, the cell is still performing its functions as per usual, but will not participate in the cycle itself, which is growing and duplicating. This transition from the main cell cycle into G0 may be permanent as cells differentiate, for example, neurons, or temporary, such as differentiated liver cells that will enter the cell cycle if needed. Abigail will not be entering G0 as she is a stem cell. With the negative regulator requirements met, transcription factors are released and new proteins are made. Abigail now looks towards the positive regulators. Cyclins are a group of proteins that promote the continuation of the cell cycle when its conditions are met. There are 4 major cyclins that come into play, namely Cyclin D or the G1 cyclin, Cyclin E or the G1/S cyclin, Cyclin A or the S cyclin, and Cyclin B or the M cyclin. Currently, Abigail has produced only 2 cyclins, which are Cyclin D and Cyclin E. First, after RB allows the production of cyclins, Cyclin D will help Abigail proceed past most of the negative regulator requirements. Once that happens, Cyclin E will start being produced to initiate the production of DNA replicating enzymes. Note that all positive regulators are also proteins and are encoded by proto-oncogenes. Finally, after all negative regulators are inhibited, all preparations for DNA synthesis is ready as prepared by the positive regulators. Abigail is ready to transition into the S phase. DNA polymerase will begin adding nucleotides to a DNA strand separated by helicase, held separated by single-stranded binding proteins or SSB proteins, prevented from coiling too much by topoisomerase, at a single starting point signaled by a primer, which is an RNA enzyme constructed by primase and will proofread once done. Due to the intrinsic nature of the orientation of DNA, which are the 3' and 5' ends, and due to the fact that DNA polymerase can only build from an anti-parallel position, out of the 2 strands of DNA, one will have a smooth traverse and another one will have to get resignaled by primase to move up the strand as helicase separates more and more DNA. As a result, they are called the leading and lagging strands respectively. In the lagging strand, each primer-induced DNA fragment is denoted as an Okazaki fragment and will have to be glued together by a ligase. Furthermore, each primer in the lagging strand and one primer in the leading strand will have to be converted from RNA to DNA. Now that DNA replication has finished, Abigail is yet again faced with another question. Was DNA replication successful? Multiple processes in Abigail will review DNA polymerase's job and determine if there are any problems such as breaks or errors. There are multiple ways of detection of faulty DNA, and the most prominent one is the detection of residue SIR198-1, a type of antibody. The negative regulator ataxia-telangiectasia mutated serine threonine kinase, or ATM kinase, will become autophosphorylated and signal for a halt in the cell cycle if it detects DNA damage. Afterwards, it will recruit a family of enzymes including breast cancer type 1 susceptibility protein, or BRCA1, to mediate DNA repair or apoptosis if the DNA damage is too excessive. Now, without further ado, Abigail will proceed to the G2 state. G2 in a nutshell is basically where the cell duplicates all its required organelles and do one final check before mitosis. Necessary meiotic proteins are produced and Abigail is again in a similar situation with the negative regulators as in G1. Ataxia-telangiectasia and RAD3-related serine and threonine kinase, also known as ATR, along with ATM, will examine DNA once more, repairing any damaged sites it can find. Checkpoint kinases 1 and 2, or CHK1 and CHK2, will also be produced, along with the resurgence of P53 from back in the G1 checkpoint. The concentration of cyclin A will rise, resulting in the duplication of centrioles, which are organelles constructed out of bands of 3 microtubules and other organelles. The rise in cyclin A will also signal the rise in cyclin B, which will dissipate microtubule-constructed cellular skeletons and form maturation-promoting factors, or MPS. This will induce spindle formation, breakdown of the nuclear membrane, and the condensation of DNA in the form of chromatin to chromosomes, which link together by a centromere. Abigail is now done preparing and will begin mitosis. Abigail will cease all actions and solely focus on division. Mitosis has within itself 6 different phases, which are prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. Let's go over them one by one. During prophase, Abigail will finish chromosome condensation and the partial dissipation of the nuclear membrane, which removes the nucleus temporarily. Note that a chromosome is two copies of a part of DNA called chromatids connected together by a centromere. The centrosomes, constructed out of microtubules and now a pair of centrioles from prior G2 duplication, will proceed with the construction of the meiotic spindle and move to the polar ends of Abigail, pushed along by motoproteins. The centrosomes will polymerize the protein tubulin to construct the spindle. Next, Abigail will enter prometaphase. The nuclear membrane will be completely broken down into small membrane vesicles and the centrosomes. Now, at the opposite ends of Abigail, will send microtubules to attach themselves to proteins that are embedded within the chromosomes, called chromosomal kinetochores. This will help later to pull apart the chromosomes with the use of ATP. Entering metaphase now, Abigail's centromeres will align all the chromosomes along an equatorial plane. Since this is a crucial step in mitosis where all the chromosomes must be separated correctly, Abigail will begin the spindle checkpoint. Abigail will check for two things: whether or not all chromosomes are attached, and if so, are they attached correctly. Now also remember the MPF complex arising from cyclin B from G2? Well, when entering mitosis, MPF will trigger its own destruction by activating anaphase promoting complex/cyclosome, or APC/C, during the metaphase checkpoint. First, however, the protein meiotic arrest deficient 1, or MAD1, will have to verify all chromosomes are attached correctly, then it will deactivate and allow APC/C to take over. APC/C will tag CDK cyclin B complexes with ubiquitin, or UB. The cell will send the tagged complex to a proteasome, which is a specialized type of lysosome that only trashes UB-marked molecules. The destruction of cyclins allow future daughter cells to return to the G1 phase. However, APC/C's main job isn't exactly at destroying cyclins, and instead will also destroy the protein called securid, which inhibits separase. Once separase is released, it cuts cohesins, destroying the centromere and sister chromatids are separated. Once the chromosomes are separated, Abigail enters anaphase. Microtubules connected to the chromatids shorten, pulling the chromatids along with it to the polar ends of the cell. Then, another set of microtubules will push against another, causing cell elongation. Then, Abigail arrives at telophase. During telophase, the aforementioned second set of microtubules will continue to elongate, stretching the cell even more, while a nuclear envelope forms from the fragmented vesicles from before. Once the nuclear envelope reappears, chromosomes will then decondense into chromatin, while centrosomes will decommission and disappear. Finally, Abigail has one last thing to do: sever connection between the cellular membranes, or cytokinesis. Cytokinesis is actually not a phase of mitosis, but for simplicity, we are going to lob it into the end state. The Golgi apparatuses of both daughter cells will send signals down to the place of separation, inducing a contractile ring resulting in a cleavage furrow. Then, the microtubules contract and separation is initiated. Voila! Abigail is now no longer Abigail, but two separate cells, which we will denote to be Abigail 1 and Abigail 2. Now, in theory, both Abigail 1 and Abigail 2 are the same as the original Abigail. They have the same set of DNA, they have the same set of organelles, and etc. Unfortunately, no. Although 3 billion years of evolution has made our biological DNA replication a good process and our cell environment efficient and safe, it is nowhere near perfect. DNA replication is good at the middle sections but are prone to mistakes at the ends. Even if there were no mistakes, reactive oxygen species or other detrimental factors will erode and nibble at the ends of DNA, making the ends extremely unsafe. To combat this, evolution has made it that every cell that undergoes an unfixable problem go into apoptosis, but sometimes even that function is damaged. And when even the immune system's defenses, the natural killer cells, or NKC, and cytotoxic T cells, or CD8+, etc., fails to fix this, cancer is the result. Evolution has also given us a telomere, which is a long sequence of repetitive strands of DNA that cap the ends of chromatids. As a cell replicates, the telomeres tank the DNA damage that would have occurred to very important DNA and will shorten over time as a consequence. As telomeres are very necessary for DNA integrity, after 40 divisions or so, telomeres will have become so small that the negative regulators in a cell simply refuse to allow a cell to go past the G1 checkpoint. Despite this, there is not enough DNA damage for a cell to undergo apoptosis, so the cell would just sit there, not going to G0, stuck at G1. This is a senescent cell, a cell that will no longer divide nor undergo programmed cell death. And as one ages, these cells will accumulate and cause the degradation of bodily function. Similar things happen to organelles and of the like. As a cell divides, it will reach a point where an organelle is just exactly not copied right, and the resulting daughter cells will have attained organelles that are broken or inefficient at their jobs, making processes and productions that are crucial to a cell slow or even non-existent. And now we have arrived at the end of the cell cycle. Let's have a recap. The cell cycle has 3 states: interphase, resting, and mitosis. Interphase consists of 3 phases, G1, S, and G2. Each phase has its own constituent positive regulator, the cyclin, which binds to CDKs to act as a positive regulator. G1 is when a cell prepares to divide. It is regulated by the negative regulators RB and P53. A cell may choose during early G1 that it will exit the cell cycle either by differentiation or insufficient resources and will move to the state G0. S is when a cell duplicates DNA. It is regulated by the negative regulator ATM. G2 is when a cell continues to prepare for mitosis and is regulated again by P53. The state mitosis contains 6 phases: prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. Cytokinesis is technically not part of mitosis, but we will include it for simplicity. Prophase is when chromosomes condense. Prometaphase is when the nuclear envelope is fragmented and the meiotic spindle is formed. Metaphase is when all chromosomes are aligned at the equatorial plane and the cell performs the spindle checkpoint to verify such. Anaphase is when the chromosomes are separated. Telophase is when the nuclear membrane redevelops, and cytokinesis is when the 2 daughter cells sever membrane connection. Of course, the cell cycle is a complicated biological process, and even now, we are discovering new proteins and processes that regulate it. By no means is this overview a complete one, as biologists continue to find new things. However, I hope my video helped you give a basic overview on the cell cycle. Thanks for watching.